It's all yours, SFT.
Thanks so much. Thanks so much for joining us, guys. This is Standing for Truth. We got a good discussion tonight. I've got SpeedOfSound here, who joined us just a few weeks ago, I believe. We had a debate on genetics and whether it is evidence for creation or evolution. So that got a lot of good feedback. If you haven't seen it, please check it out. Today's topic is human chromosome 2 fusion evidence for evolution. So this is going to be a lot of fun. It is an informal discussion, just back and forth. We're going to keep it respectful. If there are some questions from the audience, just tag Standing for Truth. We'll save them when the discussion part's over. We'll try and get to some of the questions from the audience. So once again, thanks so much for joining, everybody. Mike, thanks so much for doing this. I'm looking forward to this. So since you're the guest, unless you want to say a few words in terms of introduction, you can go ahead and do that. And then just kind of go right into, why you believe the evidence for the chromosomal 2 fusion points to evolution. Go ahead, Mike.
Okay, so there's two parts of this. And if my audio is not okay, I hope somebody lets us know. There's two parts of this. There's a meta part that basically, if chromosome 2 fusion did happen, what does that mean for evolution? And the important thing to note there is that it has never been considered a slam dunk proof for common descent for chimpanzees and humans. It just removes basically one of the biggest problems for that evolution. If the chimpanzees had 48 chromosomes, we have 46, then we have to wonder why. Chromosome 2 fusion solves that problem for evolution. So rather than being offered as proof for evolution, which is kind of an odd idea for, as far as I'm concerned anyway, it removes a roadblock and it makes the story good. And overall, each of us have a story. You have the story of Adam and Eve, and you derive basically what is happening now from that story. So between then and now, we have to figure out what happened. So that's kind of what we're both doing. Evolution has a story that there was a species Y that both chimpanzees and us descended from, and this chromosome 2 fusion fits in with that story rather well. And of course, it also fits your story. God could have smushed 2 chromosomes together, or there could have been a chromosome 2 fusion after Adam and Eve. So I mean, it isn't proof one way or the other, I think, is the point. And the second part is the details of Sanford and his criticism of chromosome 2 fusion, which I find to be weak, very weak. There are quite a few problems, so we can. I'll let you go, and we can get into it. It would be cool if I could screen share. But for now, what do you have to say?
Yeah, if we get to a point where you want to screen share, I think that is possible. Is that right, Praise? Mike could screen share at some point?
Yeah, absolutely. It's you should see in the bottom of the screen.
I do.
Okay.
Yeah.
And I guess regarding everything you said, I would agree. It is invoked, this chromosome 2 fusion, to explain the differences between chromosomes in humans and the great apes, right? We're looking at the great apes, which have 48 chromosomes, and humans have 46. So technically, evolutionists, right, they claim that this human chromosome number 2 arose by the fusion of 2 chromosomes in this theoretical, hypothetical chimp human common ancestor, right? They say that it arose after the splitting, kind of like what you said, after the splitting of the human and chimp lineages from their common ancestor. So it would have happened in the human line. So like you said, it would be indirect evidence anyways, because yes, we could explain it within our biblical base model, right? Post-flood or pre-flood, since we do believe in a bottleneck, it could have easily been passed down. But that fusing ape chromosome, the two ape chromosomes, I should say, they were previously known as chromosome 12 and 13. Now the evolutionists call it chromosome 2A and 2B. So that's how sure they really are that
They're
this
very sure. Yeah.
occurred, right? They say that these two chromosomes merged to create what we now know of as human chromosome 2. And just like you said, that's how they deal with the differences in human and ape chromosomes. But I will start off by saying fusions do happen in nature, and the resulting fused chromosome, Mike, it can be transmitted to the next generation. And I don't think anyone's going to disagree with that. So fusions do happen. But I think this goes back to the fact of genetic degeneration, because those fusions are generally very deleterious. I'm not sure if you would agree, but they do reduce fertility, and they do disrupt chromosome structure and chromosome function. So normally there should at least be selection against those types of events. I would say in general, if there was a fusion in the human line, fusions in general are manifestations of genetic entropy. So that would certainly be consistent with the fall. But when it comes to germline fusions, because that's what this would have to be, Mike, obviously, a fusion that's actually passed on, they're not normally, they don't normally arise as end-to-end, telomere to telomere fusions. Usually it's due to mistakes in the crossing over process. So like, what kind of fusion do you believe this was? Like, was this a telomere to telomere fusion, or was it a fusion that occurred due to a mistake in the crossing over process? Like, what do you believe in regards to that, Mike? And take your time. Give us your thoughts on that.
Okay. First of all, I don't feel totally prepared on chromosome 2, but I decided to go with this anyway. But yes, telomere to telomere fusion. One of the strands got broken, and the other one, there are a number of examples. I'm not sure. That's the part that I would like to know more about, is these telomere to telomere fusions. They are possible. They do happen. And I understand that 1 out of 1,000 human beings are born with some sort of a fusion. And there are a number of different ways for it to happen. There's no reason why it couldn't. Fertility is decreased. It's obviously because there's a different number of chromosomes, but it is possible. There's I understand that there's at least one family in Sweden that has like 9 generations where the chromosome fusion has actually made it into the progeny. So it's all possible. There's no reason why that can't happen. And we have a number of
I'm sorry, I just want to clarify. Is that fusion you just mentioned? For one, I'm guessing it's documented. 2, is that due to a mistake in the crossing over process, or is that a telomere to telomere, like a head-to-head fusion that is being purported in the human chromosome 2 fusion? Like which type of fusion is it? Do you know?
I don't know. I haven't checked that one.
Because, and that's okay. That's okay. And I don't know either. I'll just have to look at the documentation on that. Because from my understanding, Mike, most, and I always say the majority proves the rule. So I could be shown a few in contradiction to what I'm about to say, but most documented mammalian fusions, and I'm guessing this goes for the specific case you just mentioned, involve specific categories of sequences that are called satellite DNA. And that satellite DNA, Mike, it's found throughout the genome, and particularly around centromeres. right? Like you were saying earlier, where most centromeres break prior to the actual fusing. So that presence of this satellite DNA is actually a very key genomic feature that comprises the initial breakage and then subsequent fuse sequence observed in all studied fusions found in mammals. But the problem here that I'd like you to address is the fact that this purported fusion site has no satellite DNA, which is contradictory to all other observed mammalian fusion. So that's just one of the, I guess, inconsistencies to this so-called hypothetical fusion. Like, are you aware of this satellite DNA inconsistency?
There's plenty of examples of telomere to telomere fusions, particularly in the somatic line, like in cancers and a number of mutations in yeast. I mean, it's common, so it's really not a roadblock. It's just a different kind of fusion. And as far as how common it is and all that, that's part of my unpreparedness. I have not checked that part of it, but I did look into a few other things, some of Sanford's criticisms. So we,
right. If I could just mention like, for example, with the cancer
right.
fusions that occur. Yeah, you're absolutely right about that. That does occur as telomere to telomere fusions. But the thing is, those that occur in cancer cells, those aren't actually passed on, right? No, so this one,
somatic.
Right. So, and that's kind of why I specified earlier that it would have to be a germline mutation, right? So technically, this satellite DNA that would need to be found in the human chromosome 2 fusion, from my understanding, I think it was in 1991, because first they recognized it based on banding patterns in chromosomes, right? They saw these similarities between some human and ape chromosomes, but I believe it was in 1991 when they actually discovered the so-called fusion site, but wasn't exactly what they expected. It had a totally different pattern than what is known and found in living mammals, right? Not the cancer fusions, no, but in the cancer cells.
They have found these. They have found these examples in other, and that is not a roadblock. I mean, it's a rare event, but just because it's rare does not mean it didn't happen. Okay?
Right. Well,
there's no ultimate hard roadblock to this happening. It does happen, and we've witnessed. We have examples of it. And
well, I guess I'm just treading lightly because I've looked thoroughly and I haven't seen any examples of a telomere to telomere fusion that is being purported in the one in the human line, right? Because this one in the human line, the human chromosome number 2, it is a fact that it didn't contain this satellite DNA as is found in living mammalian fusions. So because normally, Mike, what they find is satellite DNA combined with satellite DNA or satellite DNA combined with telomeric DNA. But the telomere to telomere fusions is rarely, if ever, heard of or observed. Now I'm just treading cautiously because maybe you could point to one or two, say after the debate, maybe you'll show me a paper. But yeah, it is a very rare fusion. And some have even said that if this really was a fusion, it would be the first of its kind observed in nature because of this lack of satellite DNA being seen. But and I want you to comment on this one because I don't want to beat a dead horse. Is the thing is you should see the telltale signs of this remnant telomeric DNA, right? Those sequences. But right. What they actually discovered, and I'll give you plenty of time to comment on this, Mike, is that the telomeric sequences were very, very small, right? As you know, human telomeres contain, I believe it's about 10,000 to 15,000 bases in that sequence. So if this was really a fusion and we're looking at it and we're doing our due diligence on this fusion, we should be seeing at least 20,000 to 30,000 bases lengthwise. But this fusion site, you may know about this, Mike, it's only about 800 bases in length. And here's the key point to consider. It's only about 70% similar to what would be a pristine fusion, and it's highly degraded. So how do you explain the lack of remnant telomeric sequences that should be found if it is a telomere to telomere, head to head fusion? Go ahead, take as much time as you need.
Okay, the fact that the telomeres are short is one of the reasons why it fused. That's
so. Are you?
Go ahead.
Are you hypothesizing that it was degraded? Because as we know, telomeres being the end caps of chromosomes, they're supposed to protect from fusion. So yeah, my question would be, why was there a fusion, and why were the telomeres involved when their primary job is to prevent fusion? So are you trying to say that? Are you hypothesizing that then these telomeres were shortened and degraded, therefore that's how the fusion occurred?
Right, there'd be a remnant telomere on one side, and the other side would be some sort of a breakage in one of the chromosomes, and that's where the fusion happened. So we wouldn't expect to see a whole lot of telomeres. What we see is exactly what we'd expect. Can I share my screen?
Yes, yes, and yeah, of course, share the screen. And I understand what you're saying. Okay, so I'm glad you do agree that the fusion site is only about 800 or less bases in length, and about 70% similar to what a pristine fusion should be. That it is highly degraded, because I've had evolutionists disagree with that. For example, in my debate with Erica, she disagreed with that, or I don't know how familiar she actually was with the data. She might have just been using talking points. But here's the thing, and I understand what you're saying, okay, that according to you, that's what you'd expect. Now, here's the problem, okay? These internal telomeric sequences that are actually found in very small numbers, when I do believe that it should be found in excess of what it is. But it turns out that those internal telomeric sequences are found all over the genome, Mike. And there's been many papers, I can send them to you, that have been published that are giving us functions in many of these sites. For example, they're binding to things called transcription factors. And that, as you know, initiates the copying process of a gene into RNA. So those sequences being seen are not the remnants of telomeres from a past chromosomal 2 fusion. They're actually just functional sequences that are found all throughout the genome. So how would you explain that?
Okay. I did a search on chromosome 2, on the entire chromosome 2 for the TTA-GG sequence.
Right.
GGG. Very few of them. It randomly occurs because of the shortness of that thing. It's always going to occur, and you're going to find them all over, but you never find more than one. When I did a search on chromosome 2 for 2 in a row, the only place I found it is at this telomeres fusion site. And I got it on the screen right here. I'm not sure if you can see that.
Yeah.
It might be too. Okay. Right here's the fusion site. And I sort of broke them up into five base pairs. There are a couple of indels, and then it goes on, and then you see the reverse ones down here, and it's not very big. And then I went out and I started searching for the exons, and I found that there are 4 exons. Well, okay, that's another subject. But this fusion site is here, and there's nowhere else in this chromosome where I have found anything as frighteningly similar to telomeres as right here. Right smack in the middle at 14.1 on chromosome 2, right at the end of that DDX11L2 gene, which you see up here. Okay. And this is it. And
it's a very short sequence though, Mike. And what I'm saying is those internal telomeric sequences that you're pointing to, I agree with that. For one, they're highly degraded. They're not reflective of what a pristine fusion should look like. And as I've just indicated, they're not the remnants of a past chromosomal 2 fusion, they are playing important roles in the genome, and they are found all over the genome. Are you pointing to the so-called remnant telomeres here with the sequences that you're pointing to?
Yeah, it's this section right here. I don't know if you can't annotate on this. Probably about, it looks like about maybe 600-700, something like that. In this section. The thing is, how many telomere to telomere fusion sites have you looked at?
Are you saying how many telomere to telomere chromosomal fusion events have I looked at in nature?
Yes. How many have you pulled up on NCBI and looked at?
Well, exactly. So what I've found is chromosomal fusions based on mistakes and errors in the crossing over process. I've seen telomere to telomere fusions in cancer cells, but those aren't passed on, like you said correctly. Those occur in the somatic cell line, so they're not passed on. But the actual ones that are passed on have never been observed to be the type of fusion that is being purported in the human chromosome number 2. And the thing is, these bases being extremely degenerate was not a prediction or expectation, Mike.
Oh, yes,
by the discoverers of the human chromosome 2.
It really is. It really is expected. We have 400,000 generations. Okay, and there's you see the TTAG here, TTAGG. There's a TTTGG. There's like one base in each of these things that are off. And down below, it's the same thing. I mean, it keeps repeating over and over again. There are mutations here, and we would expect there to be mutations here because this is not a conserved region of DNA because it doesn't do anything. So,
well, isn't the fusion site located in an area of the human genome with low levels of recombination because it's near a centromere? Like, that should mean that the mutation rates in that area of the fusion site should be extremely low. And if that fusion did occur, let's say it did occur, Mike, there should actually be very little degeneration. That's why I'm trying to say that they expected it to be fairly pristine. Would you agree that it's only about 700 to 800 bases in length? I mean, that's a pretty extremely tiny signature for such an event. How long ago did this happen? Because you just mentioned the amount of generations. Like, when do you believe, or what does the evolutionary literature say this fusion event occurred? Like, was it hundreds of thousands of years ago, or was it millions of years ago?
I'm guessing it would be millions of years. Well, it would definitely be millions of years ago, probably around 6 to 8 million years ago. So so that'd be 300,000 to 400,000 generations. This is a perfectly normal amount of mutations to see. Recombination has nothing to do with whether or not something mutates. There are mutations all over, all of the time. And the only reason that you don't see mutations in conserved areas is because the embryo dies before it forms, or the critter dies before it even gets out of childhood. So,
well, real quick before I comment again on those telomere repeats, because I am genuinely curious. Let's say, so we got 6 million years ago, roughly, the evolutionary literature says. Then the human line and the chimp line split. Okay, so somewhere after 6 million years ago, whether it's 4 million, 2 million, I have read possibly even 800,000 years ago, that this fusion occurred. Okay, so let's say it occurred in a hypothetical population within that time frame, between 800,000 to say 4 million years ago. How did this fusion, okay, how is it transmitted to everybody on planet Earth? For example, every human today has 46 chromosomes. But when this would have occurred, you would have had, what, one person with the fusion, okay? But everybody else would have 48 chromosomes still. And I'm guessing not everybody was in the same area at the time. Like, how was it passed on so everybody today has this so-called chromosomal 2 fusion?
Genetic drift. It would drift into the population. It's the same thing with Y chromosome atom. It's the same reason why you could trace back and find common ancestors every so often. Common ancestor of everyone in a tree because that is just the way genetics works. If you've ever run any models on a computer, you'd see that frequently all the lines die out except for one. You can trace it back to one ancestor somewhere along the lineage, and there's all kinds of Y chromosomal atoms. There's going all the way back. Just the way genetics works. But let's get back to, like, you're having a problem with the way this fusion site looks, that it's too small. But the thing is, what do we have to compare with? We don't know. It's basically that you don't like it.
Well, it's hypothetical. Like it. Well, I'm saying it's hypothetical theoretical because it's unlike any telomere. It's unlike any fusion we've actually observed in nature. And my rebuttal to those internal telomere repeats that you're pointing to, I see them loud and clear. There's no denying it. But I'm saying that the presence of those internally located telomere repeats, Mike, they occur, and this is a fact, and I can send you papers proving it. They occur throughout the human genome from only a few repeats in length, like we see there. And I'm glad you actually shared screen so the audience can see, to literally, Mike, hundreds of repeats long. And telomeric repeat sequences are unique to the ends of chromosomes, but they're distributed widely over the human genome with function. So I don't know why we keep asserting that this.
I searched the entire human chromosome 2 is very big. It's one of the big ones. I didn't search all of the chromosomes, but I did 2, and I only found one place where there's 2 repeats of this 6 base sequence that indicates a telomere. So if you have evidence of this appearing you're saying that there are telomeric remnants all over the genome? Where?
No, I'm saying that those aren't telomeric remnants. I'm saying that those sequences you're looking at, those internally. See, the thing is, you're looking at a very, very degenerate. You're looking at something that's only between 700 and 800 bases long. Telomeric DNA is what, 10,000 bases long. So this is very small. You've admitted this, and I understand your.
But
your
it's not
degenerate. It happened a long time ago, right? You would expect this, but. The thing is, these internal telomeric repeats you're pointing to, I'm saying that they occur all throughout the genome, not just in this human chromosome.
No,
it is a fact, yes, and they contain, and they're functional. You're seeing more and more. Okay, so if I can show you after the debate that these internal sequences are found throughout the genome, and they're not just unique to this chromosome.
And you're saying that they're not in telomeres, that they're somewhere embedded in the rest of the chromosome. Yeah, they have found a few. I just read a paper, and what these indicate is ancient fusion sites that go even further back, other chromosomes that fused. And they found these all over the place. They found these fusion events in a number of species, and now they're looking at these ancient remnants. And there are a few of them, but in chromosomes,
assuming ancient remnants.
What's that?
I'm saying they're assuming ancient remnants because the fact
is like evidence for an ancient remnant of a chromosome fusion, and enough to alert them. But
here's the thing, Mike. Real quick before I lose my train of thought on this one, I understand these sequences. I agree with them. I'm trying to say that they're very small, they're degenerate. But the fact that this short sequence does have some very limited homology to, say, telomeric DNA.
No, it's not limited. It's extremely clear. I mean, I can see it. I'm reading the repeats over and over again. And there are mutations, which is to be expected. But this is a very clear telomere.
It's clear. It's clear that they're degenerate. It's clear that they're small. It's clear that it was a surprise when they discovered that it was so degenerate. And it's also a fact that these telomeric-like motifs are found throughout the genome with function and do not show any evidence of being ancient fusion sites. That part is just hypothetical. But if you're so sure of this, answer me this, because we're talking about telomeres here. We're talking about the telomeric sequences. But if there's any validity to this, okay, the fused chromosome not only requires the observation of these remnant telomeres, Mike, it also requires demonstration of a legitimate fusion site, but also a disabled second centromere, right? The cryptic centromere, as they call it. But what they point to, Mike, is 171 base pair repeat that they say characterizes the primate centromeres. But the thing is, for one, this site too is very small. It's too human to be a remnant centromere. And this is very interesting and telling right here, Mike. This entire sequence that they point to, that they say is the cryptic centromere, is internal to a functional gene. So can you point me to this cryptic centromere that would have to be found if there's any validity to what you're saying here?
My unpreparedness did not allow me to find that centromere. So that's part of the research I haven't done yet. But I would challenge you to provide me with a paper in the comments of this debate that shows me that there are long repeats of telomeric repeats that are functional somewhere out in the genome. I need to see those papers. You can't just pull information after your last video with Erica, which I'm hoping we can talk about, with the paper that you put up that was about gene duplication. I want you to show me that paper. Otherwise, we're going to have to ignore it. I mean, I can't believe.
Right. Well, let's not divert the topic. And we will, Mike. I'm saying,
but there's more to this. There's a fusion site here, right? I mean, if that was the only evidence we have, that would be pretty damn weak. When they found this fusion,
and that's why I'm pointing to the fact that remnant centromeres are difficult to find. The remnant telomeres are difficult to find. Why are they so degenerate? I understand you explained them because it happened hundreds and thousands of years ago to millions of years ago. But the fact is, the telomeric repeats that are seen throughout the genome, and I'll prove to you that later if you
have you actually looked at
testing that. But here's the thing. There's also no satellite DNA that's being found. Okay, and what's most important is the fact that this site, okay, this fusion site is in the middle of what you know as the first intron. And I'm sure you're aware of the argument from Tompkins.
DDX11 or 11L2.
Right. So I just want to, because you said it was Sanford earlier, but it's actually Tompkins.
Tompkins, okay. Well, didn't Sanford have something? No, maybe not. Anyway, yeah, we could talk about that because I've got some information. Why did my screen share stop, or did it? Am I still? No, I'm not sharing anymore. It says I'm sharing, but it's not sharing. Oh, now it is. What the hell?
I think it's...
Okay, so we have this right here now. So I went out and I looked at, there is, let's see here. We got DDX11. This is, okay, it's DDX11.
But as you go, I just want to explain to the audience, and obviously you're aware of this, but this purported fusion site, okay, so it's not just the lack of remnant centromeres, the degenerate telomeres, which I explained. I know that you at least had a hypothesis for why those are so degenerate. The lack of satellite DNA, the fact that it is indirect evidence anyways for evolution, because like you said, it could have occurred in the human line according to the biblical base model, but that purported fusion site is located inside a highly expressed gene. And that purported fusion site, Mike, also encodes a region inside the DDX11L2 gene, like you're talking about, where transcription factors bind. And I can go on and on and on regarding the...
But you're looking at it right here. This is your highly expressed gene, which is not highly expressed at all. Like in, let's see, over here, one of the higher ones, 5 parts per million. Over here in the testes are kind of like they'll sleep with any gene they express. And even this one only has 18 parts per million. This is not highly expressed. If you look at the actual DDX11, I think this is DDX18. This is DDX11. This is the DDX11 gene on chromosome 12.
Well, Mike.
We have much higher expression.
It's that fusion site, Mike. It's located smack dab in the middle of a complex and functional gene, but its sequence contains a functional transcription factor, a transcription factor binding domain, and it's uniquely positioned to control the transcriptional complexity of the DDX11L2 gene. So I don't know why you're saying that it's barely expressed
I can tell you
or
why.
barely functional.
Because it's barely expressed. Okay, number one, there are 28 DDX11 genes in deadbox 11 genes in the human genome all over. There are plenty of really functional genes that are expressed at a much higher rate than this one. Secondly, there are 4 exons in this gene. One of the exons I thought was on the other side of the fusion site, and that one is, let's see, if we go down to, if you look right here, if you, let's see, this is DDX11. I'm going to look at expression, exon expression. This is DDX18.
Are you aware though, Mike, that the fusion site is actually read in the reverse complement, and it's a transcription factor binding site in the middle of that gene called DDX11L2? Like, are you? It codes. I know it doesn't code for a protein. That's why they call it a pseudogene, but that's just based on junk DNA assumptions. But it does code for a functional RNA molecule. Are you saying that that's not? And it's co-expressed too. So it's not just, it's co-expressed with other genes, and those genes are highly critical to the cycle of the cell. I mean, are you disagreeing with all this stuff? I think evolutionists at least agree that it is functional.
I can't find a single thing. They haven't even looked at the 3D map of this thing. It's considered, it's listed as a pseudogene. It is low, it does very little expression. Only one exon, exon 4, expressed at all. I have found nothing on it being co-expressed. I found nothing on it being functional, and I've searched papers everywhere. And on NCBI, when you pull up the gene, it shows you all the papers, right? So you can find all the places that people have talked about it. Nobody is talking about this gene except you guys. I see nothing to indicate this is functional, but it's even worse than that. Okay.
Well, no, it's not worse than that, because I'll provide. Have you read through Tompkins' paper where he talks about how that gene is highly expressed in not just one, but many important cells throughout the body? It's an extremely important gene, and it's linked to many other highly important genes. This is just one nail in the coffin, Mike.
No, it is not expressed. It's hardly expressed. This one is 2 parts per million. The testes is the only place this thing is expressed. But it's even worse than that. I have bad news for you. I went and searched, and I was under the impression, because I believe Tompkins, that this one exon was on the other side of the fusion site. And so I searched them out, and here we go. Exon 4 right here, down to exon 1 end. That is the whole gene. It's not that damn big. Well, no. Down here is the fusion site. He missed it. He made a mistake. That fusion site, that gene does not cross that fusion site, as far as I could be wrong, because
you are wrong, because that site, like,
how do you know?
Because that site, I'll give you his most recent papers in 2017. It's called Debunking the Debunkers. It's where he goes into detail on all these so-called arguments. But he shows that this site is in the middle of the gene in what's called the first intron. Okay.
Where does he show that? Debunking.
It literally overlaps from one end to the other, and those genes. Okay, these genes, these long non-coding RNA genes, they function exactly like protein coding genes. They have exons or coding regions that end up in the final RNA molecule. They have introns, as you know, they're intervening regions. We're looking at promoter regions, control regions. I mean, this so-called fusion site really is part of this highly functional gene, and you don't gain highly important genes by slamming together 2 chromosomes, Mike.
You have no evidence that it's highly functional at all. I can't find a thing about it being even functional in the, not even a little tiny bit. There are 28 other DDX11 genes.
It's kind of like when it comes to the endogenous retroviruses or the ALUs. A lot of the times, the evolutionists they're very silent when it comes to this, because their worldview would suggest a genome of junk. I mean, the chromosome 2 fusion is based on the junk DNA paradigm. But I'm saying the junk DNA paradigm has been completely overturned. Go ahead, Mike. What were you going to say?
It hasn't. No, he's straight off topic into the junk DNA paradigm. But anyway, Tompkins made a mistake. That fusion site is not in the middle of that gene. If you pull up the gene on NCBI, it shows you the fusion site, but none of this stuff is part of an exon. None of this. And I was surprised to find that today. I was befuddled, and I thought for sure I would find an exon on one exon on one side of the fusion site and 3 on the other. And lo and behold, all 4 are on one side, according to what I just pulled on the what is it, the 38. Have you looked at NCBI? Have you looked at these? Have you done this work that I'm doing? It took me about 3 hours.
I've done a lot of work on this human chromosome too, because I wanted to know, for one, about the satellite DNA.
So do you have a copy to show me where the exon is in that fusion site?
Yeah. Have you read his original paper and then also his paper in 2017 titled Debunking the Debunkers, where he goes over all the apparent arguments from the evolutionist community? I mean, most of the arguments have been shown and seen in blogs, but he addresses all these. I mean, they're not. So are you? So it sounds like your argument is that it doesn't span the fusion site. Okay, this gene.
Well, even if it did, it wouldn't matter, because that one exon is not even expressed. This one right here, can you see where my mouse is? That it's very short. Exon 1, hardly ever expressed in any tissue type. So
so your argument is it's not, so that purported fusion site, right, that is being shown by Tompkins here to be located inside a highly expressed gene barely has any expression, is what you're saying.
Can you do a screen share and show me this paper and show me where he says that, or show me where and the other thing is that he listed the sequence here, and I was not able to find that exact sequence. I'm guessing he went with a different genome. Are you? I don't know if you can see that or not, but that's Tompkins' paper, I think. Let's see, what is it called? This is by Tompkins and Bergman, the Chromosome 2 fusion model of human evolution.
Right. But have you seen the most recent? But have you seen the most recent paper where he addresses all these supposed arguments? Because he shows, just 1 second, Mike, because this stuff's technical. He does show that purported fusion site also, okay, he's not only saying it's inside a highly expressed gene, but also that it encodes a region inside that DDX11L2 gene, Mike, where transcription factors bind. I think because of your evolutionary worldview and assumptions, you guys don't look to the genome as something that is highly functional. I think you look at it as something like a junkyard, as you kind of stated in our last debate. But when we look at these things, Mike, as creationists, okay, we see the complexity of the genome. We don't need to explain them away by saying, well, it's barely expressed. I mean, these transcription factors, I mean, they're proteins that bind to DNA at specific regulatory sites. I mean, these things are important. So if your argument is that the fusion site is not located smack dab in the middle of a complex and functional gene, then we'll just have to agree to disagree and pass around some papers after the debate.
Right.
We may have to agree to disagree on that one. But if you haven't read his second most recent paper, which I haven't seen any rebuttal to, I think you need to at least do that. As due diligence, right?
His work is so sloppy that it's almost impossible to read. All he talks about is evolutionists and your worldview. And I've never read any other scientific paper that does that. And he has not made a case for this being highly functional. He has not made a case for any. He's making things up. I swear he's lying because he's getting paid to lie. The man should know better. His work is horrible. Let's try to do it based on what we know, rather than what Tompkins, rather than some paper that I hadn't heard of that paper yet. I haven't printed that one out yet, so I'm sure it'll be more of the same. Evolutionists this and evolutionists that. That's not what we're looking at here. Now, the other part before we leave chromosome 2 is you have this fusion site, right? The other problem is I went off to the right and I spotted a gene in the human human genome. I think it was DDX18. And then I went and found it in the chimp genome. And then there's the Synteny map. And then there's the cytological staining, which shows that the bands match exactly. That whole gene looks like a copy of those two genes smashed together. There is so much evidence, and that's why they renamed it 2A and 2B, because they are 100% sure that this happened.
Okay, well, don't slap and run. Don't slap and run. So,
no, it could happen. No.
Okay, no, I just want to comment I just want to comment on the synteny and the similarities, because
right. Why would that be?
Well, okay, so here's the facts. Okay, the similarities are there, okay, but they're overestimated by the evolutionists, because sequences that flank the reputed fusion site, as you have indicated, should match up with sequences, okay, near the ends of the corresponding chimp chromosomes, okay? But for one, those have not yet been fully sequenced, okay? So that's premature to base your entire argument for human chromosome 2 on parts of the genome in the chimp that have not been fully sequenced. But the region that actually contains the fusion site, Mike, it is uniquely human and absent in all ape genomes. So if you're going to look to synteny, okay, if you're going to look to this similarity and say that it looks exactly like the two chromosomes smashed together, explain this to me, okay? Chimps have very distinctive and very large satellite sequences, okay, as I was talking about earlier, at the ends of their chromosomes, but absent in man. And these large ape-specific satellite sequences should be, if this really is a human chromosome 2 fusion, they should be seen flanking the reputed fusion site on both sides, but they're not there. And this is a fact. I'm not sure if you're aware of this, Mike. The ape-specific satellite DNA is absolutely absent in the region of that purported fusion site. So how do you explain those inconsistencies? You can't just look to some similarity. You got to address everything as a whole. You got to look at the bigger picture, Mike.
What is your source of that? What is your source? Is it Tompkins?
No, that source is actually, let me see here. I'll you know what? I'll send it to you. I've got about 16 sources here of papers, so I'll just email them to you. But let's say hypothetically what I said is true. How do you explain that? I mean, you can't just cherry pick certain parts of, and you know what? I'm open to this being a human chromosome fusion because as you indicated earlier, it's indirect evidence anyways. It could have easily happened in the human line post-flood. Actually, we can explain it being passed on as a germline chromosome fusion easier than I think the evolutionists can. Because I know you said genetic drift, but our bottleneck post-flood, yeah, it could be passed on quite easily. But you guys, I think, would have
not post-flood. It would have had to be prior to the flood.
Right. But then it'd be passed on.
You had separate lineages, right? From who was it? Noah. Didn't he have like 3 sons or something, and they all went off into separate lineages? No, it had to happen prior to that. So it had to happen.
If it would have happened prior, right? If it would have happened prior to the flood, okay.
Right.
This telomere to telomere fusion.
Early. It would have early after Adam and Eve.
On, because it would be Noah's 3 sons and their wives. They're the ones that would repopulate humanity. I mean, that's when we can get into, I mean, we can save that for another debate, but that's where we get into the 3 main mitochondrial DNA haplogroups that take us back to Noah's 3 daughters-in-law. But yeah, that is a plausible way to pass on this supposed fusion. But what I'm saying, even though it's indirect evidence, I don't believe it's evidence for evolution anyway, since it's indirect. There are these discrepancies that you have to deal with. I understand you don't believe that this gene is functional. Okay, I know you don't believe that the reputed fusion site is internal to a functional gene. I understand that. But you do have to deal with the problems I'm talking about here. You have to deal with the remnant centromeres that are difficult to find. Hopefully you can look at that. And the telomeres, why they're so degenerate. And I know you gave an explanation for that, but there's also inconsistencies there too. So go ahead, Mike. I've talked a lot there. Take as much time as you need.
I can basically dig into that and come up with more evidence, but what you're seeing right here is a real DDX11 non-pseudogene, an actual functional protein. You see all these exons down here? There's tons of them. You see the expression of these exons? The one on these two are underexpressed, but most of them are extremely expressed. And if we go over and look at 11, we have nothing. Basically, I mean, this is just a weak little protein. I have no idea what it looks like folded because nobody has bothered with it. Because I'm sure it has some effect on the cell somewhere, but very little according to these. Here we go, 3 parts per million. It just doesn't matter. If you go and look at this, there's what do we have here? 22. Yeah, it's 22 parts per million. That one is not that high. DDX18 is a good gene, and this one has 102 parts per million. 40 parts per million. This is pretty well expressed, not just in the testes. So, I mean, this is what a real gene looks like.
Right,
non-pseudo gene. And basically this thing about, saying that the entire genome is function, that is your imagination. I mean, there's no proof of this. We found we're finding, I mean, every day they find some new mechanism in the genes. You talk about epigenetics. It's an unbelievable field. Things that are epigenetic. And there are many, many effects that we haven't unwound yet. The thing is a mess. Like I said last time, it was spaghetti code. I mean, it's just a mess. But
here's the thing, though, Mike. I mean, all you have to do is look back at the history of genetics. I think it's pretty clear that we speak probably less than 1% of the DNA language, okay, the genome that we're looking at here. Back in the 40s, the 50s, they used to say 1 gene, 1 protein, 1 function, okay? They didn't understand the genome nearly as much as we do today. So many things in science, generations later, has changed. I mean, you can look at things, Mike, in textbook generations ago and textbooks today and just see how many things have been completely overturned. We can talk for hours and hours about the massive amounts of literature that talk about all these unexpected functions of RNA.
Right.
It's like, it sounds like you agree with me, Mike, and it's amazing because
but you're betting on something that hasn't happened. I mean, we find a few pseudogenes that are functional, and you're saying that the trajectory is that we're going to find. I mean, there's 90% we have not found any function for yet. I mean, we find a few things, few surprises here and there, but your trajectory does not make
over
any
88%
sense.
of the genome. Over 88% of the genome has been discovered to be actively transcribed into RNA, which suggests function. Mike, we're dealing with a foreign language. Well, just 1 second. We're dealing with a foreign language here, okay? A very highly compressed DNA code, and it's far beyond the best computer code that we have. I mean, we write books, for example.
No.
We write books, Mike, that can be read both forwards and backwards because DNA can. Have you heard of protein moonlighting, where these mitochondrial DNA proteins, we now know, they function in this particular enzyme complex, and it takes this chemical and it's going to transform it into this or whatever. But I mean, we've got this moonlighting, and these proteins are doing things that we never, ever imagined. It's like they've got multiple functions. It's like a multifunctional tool. Like that's these proteins. It's so beyond what they used to talk about generations ago with this 1 gene, 1 protein, 1 function thing. So I think it's premature on your end to say that the genome is highly dysfunctional, because these paradigms seem to be overturned more and more and more every single day. I mean, look at the DNA redundancy, Mike, for example, right? Because there's redundancy in DNA coding for a protein, right? This wobble position 3, for example. But now we see that those play important functional roles as well. They don't just tolerate error in the cell, right? They're not just used as backup codes, but they're actually used, these redundant elements here. What they do is they slow and speed the process. So, I mean, it really is information compression on every single level. So I'm just saying, according to my basic assumption that, the Bible's true, Genesis is true, I would expect a fully functional genome. And that's why these things and I mean, this might have to be saved for a separate discussion, but like pseudogenes, the endogenous retroviruses, the chromosomal 2 fusion, I see that as evidence that evolutionists used based on an assumption that the genome is highly dysfunctional based on evolutionary leftovers. We've been going for nearly an hour here. What I want to do, since you're the guest, take as long as you want. I'm going to give you the last word to comment on anything you feel like wasn't addressed, anything that I said there. If Praise has a few questions in the comment section, then we'll jump right to that. So, Mike, I want to give you as much time as you need here. Give your closing thoughts here. Comment on anything that you feel like maybe was left addressed. Just kind of sum everything up, man. I want to thank you for joining today.
Okay. Basically, it is your worldview that's leading you to believe that this is information, or that it's language, that it's cleverly coded to do all of these things. There's a cat's ass, and that very much affects your thinking on this, and your expectation that the entire genome is going to be. And I don't like the word functional. I like the word vital, and the rest of it has a part. These are molecules. DNA is a molecule. Every single part of it is going to have an effect on the overall function of a cell, the overall health of a cell. A cell is an autopoietic system. That means self-creating. And it basically works on statistics. And all of these things that you mentioned, all these complications, all these, like the fact that the genome is read in reverse creates a little bit of RNA, and that goes ahead and blocks or promotes a gene function. These things are being discovered every day. The reason that they're being discovered every day, and they will be for the next 100 or 200 years, is because this code is not code. It's a mess. Nobody would ever program a computer like this. This is horrible. Have you ever looked at the mTOR pathway? It's ridiculous. I mean, this you're being colored by your idea, by your worldview, by the fact that you believe that these genomes were created with some perfection and that there's some clever coding going on. And what I see statistics and chaos, and I see molecules bumping into each other and creating life. And so my worldview does not allow for a creator or any kind of disembodied mind. So, of course, the only way that I can figure things out is with chemistry. The reason I believe in evolution is because of what I know about molecular biology and what I know about genetics and what I look at and how these things operate and what I know about morphogenesis. I've looked very deeply into these subjects, and it looks to me like it's all very, very possible and probably what happened. So that's about all I got. Our worldviews tend to get in the way. And now I have considered your worldview, and I intend to model it on a computer, starting with Adam and Eve, giving them as much creative heterozygosity as I can.
You got it.
I am getting tired.
You got to say it 10 times fast.
Yeah, right. So I'm basically, I am always looking for the cracks in evolution. And when I hear one of your videos, I go and investigate every bit of it because it sounds plausible. When you're really good, you're kind of like the ShamWow salesman on off his ADD drugs. Like you forgot to take your ADD drugs. You know the ShamWow salesman?
A ShamWow?
You are really good at what you do, but and you sound convincing. And when I hear you say something, it's like, oh hell, or Raw Matt, he piles stuff on faster than hell, and I have to investigate every bit of it. Every bit I've investigated has fell apart. I'm sorry, and that's kind of why I wanted to have conversations with you, because I don't see the sense in most of what you're saying. So anyway, let's try some questions. I am tired.
Right. And you know what? I want to thank you, Mike, for once again going over an hour. I mean, it's hard to believe that we can talk so much in just discussion-based format, right? So it's been fun. Obviously, I would have many things to say to what you said, but you know, as I've iterated, you're the guest here. I want to give you the final words. I think it was a really good discussion. Maybe we can do more. The first one was basically on genetics and creation versus evolution. Today we talked about human chromosome 2. Is it evidence for evolution? Maybe, you know, when we decide to have a third discussion, since you said you are open-minded to the biblical-based model, maybe I'll just put myself on the hot seat and we can talk about post-flood speciation. We can talk about created heterozygosity, and you know what? I can sit here and answer all the questions for you and see if I can convince you. And you can, in the meantime, come up with some questions you may want to ask me as well. So once again, thanks so much for doing this. It's been a great discussion. Audience looks like they've had fun. I think both our debate / discussions have been good. A lot of good feedback. We've touched on a lot of topics. I know it's been technical, so anybody in the audience is confused over anything, either email myself or Mike. Mike, there is someone in the chat named Athena, who has a channel, who really wants to get into contact with you. And I understand that you're probably too tired today, but he does want to, he holds to the evolutionary worldview. So he does want to talk to you, whether or not he wants to have you on his channel at some point. So I'll shoot you his email after this. You guys can communicate.
Feel free to share my email with Athena.
Awesome.
I saw that request before, and I actually put it in the comment that I wanted you to give my email to that person. So
awesome. I will. And then we'll, yeah, like I said, we'll coordinate another debate on a different topic at some point. If you do have a few questions.
No questions tonight, but we did have a super chat from gosh, let me hold on. I'm gonna look up real quick because I forget the names. I'm so stupid. Ashley, gosh, Ashley sent a super emoji. So for $10, that's awesome. Thank you,
awesome.
Ashley.
Thanks so much, Ashley. Thanks for supporting. And if there's no questions, I guess it was pretty fast-paced dialogue. So it flew by. Thanks again, Mike. Thanks again, Praise, for setting it up. I hope everybody had fun. And I guess we can call it a night. We'll shut her down.
Good. Always fun. Thank you.